Thank you everyone for joining us today. Let me just extend a very warm welcome to everyone. Let's just give people a few more minutes to join. I can see that quite a few number of people have joined already, but let's wait a little bit more. Just I will mention today we have a very exciting webinar, not only because of this very nice technology in cell line development, but also we have some very nice opportunities for you to acquire a device today. And we will mention this in more detail during the presentation and one-on-one. If you're interested in this technology, after this webinar, you can contact us and we will follow up happily. So I do see that still the number of attendees is increasing. I do see that it's kind of stable. I will start in a minute, probably, just one more minute. Okay, I think it looks pretty good now. Again, thank you very much everyone for joining us. We have a very nice webinar presented by my colleague, a field application specialist, Anna. She will walk you through it. But before the presentation, let's just go with some know how to use the GoToWebinar. You can see, Anna, can you move? Yeah, thank you. You can see in this slide that you can expand the control options. Also in the chat below, you can type your questions and comments anytime during the presentation. We also have two polls that will show up in your screen at some point. We will announce them, and we love to hear your answers to these questions. And without further ado, I want to give the floor to my colleague Anna.
So, hi, I'm Anna. I'm happy to take over. So thank you, Adrian, for the short introduction to GoToWebinar. So now I want to present you highly efficient cell line development workflows. And today's focus will be on the C side, so the device you can see on the right side. So just a few words about CellLink. So it's a rapidly growing company and actually also a pretty young one. So we were founded in 2016, but nevertheless, we are present in more than 60 countries and in 6 continents. We have 9 offices and the team members are over 330 right now. And also we have products and devices distributed over 1,700 labs. So you can see quite some steps over the last years. And here you can actually see some of the products we have. So you can see that CellLink quite covers a huge field of applications. So starting from bioprinters up here, so the BioAccess, the Incredibles, then we have the LumenX, HolographiX. We also have liquid volume handlers, so with the iDot Mini and the iDot. These two are actually produced by Dispendix, which is located in Germany. And then there are single cell printers down here, this one also, which are from Cytina. This is also a Germany-based company. And we also have a cell imager, so the CellSite X here. So as I already said, Cytina is a company based in Germany, so it's actually in Freiburg. And the company provides instruments for automated isolation of single living cells. and this with the assurance of clonality. So here you can see the SCP. So this is the first single cell printer, which was launched in 2015. And then 3 years afterwards, we presented the X-Site series, which comprises the C site, then the B site here with the gray lid. So this is for the isolation of bacterial cells. And then behind in the back, there is the F site with a laser integrated for fluorescent cell sorting. And the last device or product which was released is the Seabird here. This is actually a small bioreactor. So we are pretty proud that we have the world's top pharma and biotech companies as our customers. You can see quite some popular names here on this list. So with that, I want to present you the content of today's webinar. So first, I will give a short overview of the single cell applications which our customers are mainly doing. And then I will present CYTENA's single cell dispensing technology. So I will introduce the C site and also explain the dispensing and working principle. And I will also show you how the image-based assurance of clonality is provided. And then I will shortly speak about the software. And I also included a small part of the Seabird as a bioreactor. And then in the end, I will present you the key features of the C site. So the workflows which are mainly done by our customers are, for example, cell line development. So this is quite a big field, and this typically starts with transfection of cells. And then the next step is to isolate single cells. So therefore you need assurance of clonality, and then you can really select for the best producer cells, so for the best clones, which produce your gene of interest. And then you can go further on with downstream screens for quality and stability. Another workflow is the cultivation from mixed samples. So this can be done with the B site. So just isolation of single microbial cells, and then it is possible to cultivate them and also to identify them later on. And then the third application is single cell omics. So after having isolated single cells with one of our single cell printers, you can really downscale assays like RNA-seq or whole genome amplification. So with that, I want to show you a little poll which we prepared. I'm really excited about your answers to the question you will see now. So I just give you some time to answer and just wait for another 30 seconds. Okay. So, like I already mentioned, today's focus is on cell line development. So this is the main field where the CSIDE comes into action. So this is the device. It has an optical detection system integrated, which is kind of similar to a bright field microscope. And also the nice thing is that a PC is integrated. So you don't need an additional PC. So each data which is produced by the instrument will be directly stored onto the device. And then you can get it, for example, via USB. This device uses cartridges, and the cartridges contain small microchips. So if we zoom into such a microchip, it looks like that. So this here is the nozzle of the microchip. And here you see that the cell is moving towards this nozzle. And here at the nozzle end, actually, the droplet is generated. So if this droplet contains a single cell, and also this single cell fulfills all the properties you want to have, then the droplet will be directly dispensed into your substrate of choice. This nozzle here is about 40 micron in size for the CSIDE. For the B-side, we would have a smaller one. So the CSIDE is actually a benchtop instrument. It is about 60 centimeters in length and 40 centimeters in width. So therefore it fits into regular biosafety cabinets. You have no additional tubings and there are no fluids. So as you can see here, there's lots of space to work sterile. So now I want to show you how the dispensing principle works. So the CSIDE actually is quite comparable to an inkjet printer. So you have the cartridge, the dispensing chip here, and instead for an ink, it is filled with your cell suspension. And then this chip contains of a glass front side, and in the back there is a silicon membrane. And then we have a piston, and this piston is driven by piezoelectrics. And droplet ejection actually is caused by the piston moving inwards and deflecting the silicon membrane. And then a droplet is formed and there is droplet breakup. And then by piston retraction, new liquid is coming in the chip. And also the lower end, so where the nozzle is imaged. And with that technology, actually, you have very low shear forces. So we can provide high cell viability after dispensing the cells. So now you may think, okay, how is it guaranteed that just one single cell is in the droplet and will be then dispensed into your well plate? So as I already said, the nozzle is imaged and our software recognizes what is in the droplet. So let's say if you have no cells in the droplet, so 0 cell, then we have a vacuum system. And this vacuum system makes sure that unwanted droplets are removed. So in this case, pneumatic shutter is on, and then the vacuum system would remove the empty droplet. And in case a single cell is imaged and it fulfills the criteria, then the vacuum system would be inactivated, so the droplet will be formed and directly dispensed into the well plate. And the same thing is happening with the vacuum system for 2 cells and also for cell aggregates. So in this case, you don't want to have a droplet being dispensed into the well. And then also here, the droplet is removed by the vacuum system. So the disposable cartridges we have, they are of course sterile. They come single packed in a box of 10. As you can see here, this is the box in which the cartridges are packed. And like I already said, they have a nozzle size about 40 micron. The nice thing is you only need a minimal sample volume of 5 microliters, and actually you can go up to 80 microliters in total. The nozzle and the cartridges are the only parts that get in contact with your sample, so you have no cross-contamination because of this reusable parts which may cause these problems. And there are no cleaning routines. So if you think of FACS, where you have all these cleaning steps with tubings and so on. So this is not the case with the Seaside. And also because of this drop-on-demand technology, there are no aerosols. And also the FDA is recommending the usage of disposables. So I said the images, they provide assurance of clonality. So for every single cell dispensing event, the instrument would produce an image series of 5 images in total. So let us have a closer look into the image series. The first image, which would be taken before the single cell dispensing event, is this one. So the cell is quite near to the nozzle, but it hasn't entered the region of interest yet. So this is 2 droplet dispenses before the actual single cell dispensing event. Then the second picture is actually 1 droplet dispense before the event. So the cell got closer to the nozzle, and then the cell enters the region of interest. So it enters the end of the nozzle, which is the third picture, shortly before the single cell event. And then the fourth picture is the image processing overlay. So the software has detected the cell, and then the last image is the empty nozzle. So it's directly after the single cell dispensing event. So I just want to show you some highlights of our software, which is really user-friendly. You will soon recognize that. So you see here, this is how the dispensing mode looks like. And you can type in here under plate ID your experimental name, which can be also done by a barcode reader. Then you can modify your template. In this case here, it's a 96 well plate, but of course you can also switch to a 384 well plate. And then just click on the wells you need. And the template in the end, you can also save for any additional experiments or for repeatments. And you can also give comments to to dispense different amounts of cells into single wells. So like this, if you want to have like a positive control, you can dispense, for example, 10 cells in A1, and then in the rest of the plate, you can say, okay, I just want to have 1 cell per well. And then here on the right side, you see the cell camera. And this is actually pretty nice. So you can watch a real-time video during dispensing run. So you would see all the cells going through the nozzle and leaving it. And then here in the right lower corner, we have a scatter plot. And this is for the collection of sample information. So I will show you how this functions. So this is prior to the dispensing run. Just collect samples on this button here, and then the information will be given in the scatter plot. So the scatter plot shows roundness versus size, and each dot stands for a cell, and you can also click on each individual cell, and then it will be magnified. Like here, the green circle is our algorithm, and also the parameters are shown here, and you could check whether it's really a single cell or if there are maybe 2 cells sticking to each other. Anyway, with the informations you collect here, you can set your parameters here. So you can give a range of size and roundness, and this is the actual starting point for your dispensing run. So before we come to this slide, I just want to introduce another poll. I am really excited about the questions or the answers to this question. I again give you about 30 seconds and then we can go further on. Okay, good. So the next thing what I want to show you is that there is a publication actually from Genentech, and this publication used the single cell printer. So this is actually the precursor of the CSIDE, and actually already with this device, really nice results were achieved. So the group established a full automated workflow with the SCP, and the outcome was that they reached 96% single cell ejection efficiency, and they checked over 3,000 wells. And by combining the SCP nozzle images with plate imaging, they reached over 99% assurance of clonality. So this is quite awesome. And the group mentioned that the usage of SCP dramatically reduced the number of plates used for single cell cloning. So now we come shortly to the Seabird, which is bringing early suspension culture into 96 well plates. As you can see here on the left side, this is a docking station, and 3 small bioreactors are sticked onto it. So with this stack, you can incubate 3 different plates, and the whole docking station, including the control boxes, can be put into a normal incubator. So each box consists of a control box, and then there is a special lid, and this lid has specially designed tips, and the tips are of course sterile, and they are also single use. So the tips get in contact with your sample, with the medium, which is in the well plate. So you can put the lid on a standard well plate like the Eppendorf one, and then what the control box and what the Seabird does is it mixes the medium plus the cells in the wells. So the medium is sucked up, and it's also pushed back again into the well, and you have a homogeneous mixing. And also the Seabird is possible to be adjusted in the mixing rate. So you see the advantages here compared to static culture, where you have a kind of gradient. So there is no mixing of oxygen or nutrients. You have with the Seabird suspension culture with equal distribution of metabolites or anything else. And actually we saw in own experiments that the Seabird provides better cell growth for suspension cultures than having a static culture. So this was just a short part about the Seabird. Now we come back to the seaside and just look at the key features. So what I've shown was that we have image-based assurance of clonality. So the small nozzle of the microchip is imaged, and this happens for every single cell dispensing event. The Seaside is an easy-to-use benchtop instrument because of disposable, so because of the sterile single-packed cartridges, we have no cross-contamination. The single-cell efficiency is over 95%. And because of this nice technology, the cartridge and the silicon membrane and the piston, we have high cell viability, so very low shear forces on your samples. And actually for the dispensing run, it takes about 2 to 5 minutes for a 96 well plate, for a whole plate. And per 384 well plate, you need about 10 to 20 minutes. So the instrument is also automation ready, and therefore we have included many applications here. Yes. So the exciting thing is that we now have a special offer for the Seaside. So we have up to 60% off the price, and if you're interested or if you want to have additional information, of course, you can contact us anytime, for example, at info@cytina.com. Also, we have application notes available, so you're welcome to visit our website and to look under resources. We have application notes, for example, for whole RNA-seq workflow, or also about the B-side, so isolating single microbial cells. And of course, if you get in contact with us, we can also provide an individual online meeting. We can also demo the Seaside, so we have a demo room here. We can show you the device. We can also set up a dispensing run, and of course, we can also visit you. So we can do an on-site demo in your lab and also test your cell lines with the Seaside. We do application support. And if you purchase the device, of course, we will do installation, and we can also train your staff members. And the delivery time actually is under a month. So with that, I want to thank you. I have some testimonials here from customers we have, which were all really happy with the outcome of our single cell printers. And I'm happy to take any questions of you.
Thank you a lot, Anna, for this very nice presentation. I want to invite everyone to just type your questions in the comment box in the chat, and we will be happy to answer them. I do see a few now showing up, so maybe let's start right away. So I see one question. How do you make sure that cells sticking together are not dispensed?
Okay, yeah, that's actually a good question. So first, you would have to set up a good protocol. So depending on your cell line, you would wash the cells. And then the other thing is that before you do the dispensing run, you have to set the parameters, right? So you choose a roundness which guarantees that you don't have cells sticking to each other. So the more you have a round cell, the higher is the chance that it's a single cell. So you can do it with that, and also because of the image provided, image-based provided assurance of clonality, you can be sure that just a single cell was dispensed, and in case 2 cells were dispensed, then you would see that in the images which are stored onto the device, so you can check them easily afterwards. Yeah.
Okay, we have another question. What are typical cell concentrations that you recommend for single cell printing?
So what I would recommend, good question. I would choose between 0.5 and 1 million cells per ml. But in fact, you would only use about 40-50 microliters for loading the cartridge.
How many cells per minute can you clone with the single cell printer? So maybe this can be answered by telling how many, what's the time, right, for printing one plate? Yeah.
So yeah, so basically, I think for 96 well plate, if you need about 5 minutes, then it's 5 minutes divided by 96, right? And then you have an imagination. Yeah.
What's the main application of the Seaside? I think you touched a little bit on that, but there's still this question. What's the main application of the Seaside at big pharmaceutical companies?
So really the main application is cell line development. So you have genetically modified cells, and you want to isolate really single cells which produce your gene of interest, and to get a cell line approved by the FDA in the end, you need really image-based assurance of clonality, and that's why the Seaside is good to have.
Yeah, okay, thanks. We have also a question actually for the Seabird. Can you fit a Seabird in an incubator with a fixed atmosphere composition? For example, yes, CO2. Yeah.
Okay, yeah, good question. Yeah, that's possible. So you place the whole stack, the whole docking station with the 3 small bioreactors into the incubator, and so the supply of gases or temperature come from the incubator actually.
Actually, if someone is interested, can you share again the contact information and maybe the slides? So maybe we can share at the end the main contact, right? Yes, let me see what else we see. There's still quite a lot coming up.
That's good.
Let's see it's hard to pick. Can the instrument recognize and remove any dead cells or damaged cells, or how do you remove them basically?
So again, this starts with the right protocol, because you can get rid of many dead cells if you prepare your sample the right way. And then because you have all the images from us or from the seaside, you can see really the condition of your cells. So if you look at the real-time video given in the software, and also if you look at the images which are stored onto the device, you can see if you have many debris or something like that.
Yeah, this is a really good way, right? There are different gating parameters, so you can't also take it in consideration, like roundness and so on. I have more questions actually for Seabird again. How do you couple a seabird in a seaside? Do you recommend after putting a single cell to start mixing?
That's a really good question. So I guess the seabird comes a bit later into action. So you would first have your single cells in the plate, and I guess you would just let them grow a bit. And then if you have a 96 well plate, you can put a specialized lid from the seabird on the top. So exchange it with the normal lid, and then you can have your suspension culture.
See another question. Do you see problems with cell sedimentation, or how do you make sure the cell concentration stays constant?
Yeah, also good question. So we have an agitation adapter to which a pipette tip is mounted, and the pipette tip is put in the cartridge. So it ensures mixing of your samples. So no cell sedimentation is happening in the cartridge.
Yeah. So I do see still a lot of questions and very interesting ones, especially for more practical questions or really technical questions. And for the question of time, we can answer those afterwards individually. We'll be very excited to do this.
Yeah, of course.
Anna, if I may ask you to go to the next slide, or maybe we can come back to, yeah, go to the next slide so we can share again this contact information. So you can always go to the website cytena.com and you can book, let's say, one-on-one demonstration. It can be an online consultation, but it is also possible to do on-site demonstrations depending where you are. But we do have a large team in different countries, so it is possible. So we're here for you. We'll make everything possible to establish a great protocol and integrate a device into your workflow. We work with experts all over the world. We offer solutions from the small labs to the industrial scale. And with that, I want to finalize this and I also want to welcome you to our next webinars. So the upcoming events are focused on bioprinting and 3D cell culture of spheroids. And also we have a light-based bioprinting of vascular networks. You can directly scan the QR codes, and this will take you directly to the registration mode. And I just want to give you a big thank you, everyone, and see you next time.
Thanks so much. See you.